All right, everybody. Well, small class. I know people are probably studying, but this material will be on the exam in a few weeks. So got to keep grinding on it. So today we're going to talk about the respiratory system. Okay, and the respiratory system. Ooh, does, can you shut that door? It's just so loud out there. Thank you. Thank you. All right. So today we're going to talk about the respiratory system. This is basically the way that our body is going to exchange oxygen from the outside into our body and exchange the metabolic breakdown products of food like carbon dioxide out into the external world, right? And it's kind of a complicated process. And we really have to have an integrated functioning of both the cardiovascular system, which is basically the heart and the series of pipes that are going to bring the material around, and the respiratory system, because neither system by itself is going to be able to do what we need to survive, right? Without an ability to exchange material with the actual external environment, we won't be able to have the sufficient appropriate levels of gas that we can then move around the system. Oh, let's turn that off, admit. Okay, so admit. Should pause those notifications. Okay. So let's move our thing here. Okay, so we want to obtain oxygen. We want to eliminate carbon dioxide, but that's not the only thing that the respiratory system is going to do, okay? Because we have things like we need to filter out like pathogens that are coming in through the airstream, right? So we have all sorts of sort of mucous membranes, hairs that are going to sort of try to block any pathogens from entering deep into our body. Okay, we're also going to warm and moisten that air coming in. The moistening is important because that is going to facilitate the exchange of gas across two surfaces. Okay, so pretty much all gas exchange surfaces require that surface to be moist. So the lungs or surfaces are going to be moist, and other animals like frogs that are going to exchange material or exchange gases through their skin at times, or also fish, they're sort of sitting in water and they're going to be able to exchange gas across their gills, which are also wet. So you need to have a wet surface to exchange gas. And so that's another function of the respiratory system. We also use it to maintain the pH of blood. Okay, so the pH of blood is, just basically how many protons or hydrogen ions are dissolved in the bloodstream. Okay, and that really has a profound impact on how proteins are going to function, because those could protonate particular residues on top of these on top of proteins, and that can change the conformation and effectiveness of various proteins. So you have to maintain a blood pH within a fairly narrow range. Of course, we also, through this respiratory system, have developed an incredible ability to communicate with one another, right? We produce sound during expiration of our air by vibrating vocal cords. And we can, build up this incredible technology because we can all share our thoughts with one another because of the respiratory system. Then also, why do foods taste great? It's not really because of your taste buds. Your taste buds are there to recognize a few sets of, important molecules, both nutritive molecules like amino acids, that would be the taste of umami, right? Sugars, taste of sweet, and recognize potentially dangerous compounds, so bitter things. You have a huge variety of bitter taste receptors to recognize a vast array of bitter things that could potentially be toxic. But actually, the great flavors, the really like complexities of taste really require olfaction, because all the volatile compounds, when the food gets warmed up and chewed, travel up into your nasal cavity and it hit your olfactory epithelium, and you have a much greater diversity of receptors there that's going to give you the ability to really differentiate different types of food. Okay, so you've got hundreds and hundreds of olfactory receptors. So the respiratory system is involved in those sort of fine scale discriminations of flavor that you make. And finally, the respiratory system also is going to help return blood to the heart from the pulmonary circulation. Now, the overall physics of what everything is of what this respiratory system has to operate within is that of pressure. and atmospheric pressure and partial pressures and how gases move within a fluid. Okay, so a fluid can be a liquid like water. It can also be a gas like air. Okay, and in either gas or liquids, so any fluid, there's going to be a concept of pressure. Okay, now we saw this when we were looking at those little pigments that were sort of floating in a dish with the fat globules. We saw the Brownian motion of different sized pigments moving at different speeds. And the reason that they were moving, right, was because individual molecules and constituent molecules of that fluid were striking it at random directions all the time, right? And you can observe that, and that's what drives diffusion. But also, those molecules within an enclosed volume are also going to be striking the sides of that container, right? And that produces a pressure, a pushing force on the outside of that container. Now, how could we change that force, right? We could, if we had the same number of molecules here, right, and double the size of the container, well, then there's going to be more time for the molecules to be traveling before they get to the edge of the container. Your overall pressure is going to go down if you expand the size of something. Likewise, if you can condense the size of the container and have the same number of molecules, the pressure will go up because they're bouncing back and forth more often, right? Another way that you could change pressure by changing the temperature of a container. Okay, so what is temperature? Temperature is simply a measurement of how the average speed at which molecules inside a closed container are moving. Okay? It's basically the amount of energy that they contain. So if you increase the temperature, you're going to increase the speed at which these molecules are moving, and that will increase the pressure, right? Because they're hitting harder. They're hitting the surface of the walls harder. So we sort of live in this atmosphere where we're constantly being bombarded on all sides of our body by these molecules of oxygen and of nitrogen and of other sort of more rare gases within the atmosphere. And these gases move independently due to Dalton's law of partial pressures. Okay, so you can imagine here that oxygen is sort of bouncing all around like this, nitrogen bouncing all around like this. Together, their combined pressure adds up to create the total pressure. Okay. Now, any gas is going to also want to move down a pressure gradient. Okay, so if you have a high concentration of oxygen or a high pressure of oxygen in one area, you have a lower pressure of oxygen in another area, then there's going to be a tendency for that oxygen to try to equilibrate to equal the concentration on both sides. Okay? So that's going to be considering moving down the pressure gradient, okay, from higher to lower pressure. So we can calculate the overall partial pressure, which is basically just the component that one of those molecular species contributes to the total pressure by multiplying the percentage of, by multiplying the total pressure times the percentage or fraction of the gas or the fluid that is made up by that gas component. So in this case, at standard atmospheric pressure, we have 760 millimeters of mercury of pressure, and we've got about 21% of the air is oxygen. So we just multiply 760 by 0.21 or 0.209, if you want to be more precise, and you get about 159 millimeters of mercury for oxygen at standard atmospheric pressure when we're at sea level. Okay, and if you look sort of across, what are the major constituents of air? We've got nitrogen is the primary constituent. It's about 79% nitrogen, about 21% oxygen. We've got a little bit of carbon dioxide in there. It's going up, every year. And then we've got some other gases. We've got water vapor. And so together, put it all together, you get about 760 millimeters of mercury of total pressure. Now that's at sea level, okay? And our bodies are designed to, survive, and our respiratory systems are designed to survive at sea level, and probably some range above sea level where we can extract that oxygen from the atmosphere. But, if we go to Mount Everest, like we talked about in that first lecture, we've got much lower temperatures, but also we have much lower atmospheric pressure up there. Okay, just because, basically most of the atmosphere is actually pretty thin on Earth, right? If you go up a few miles, you're only, Everest is 29,000 feet up, so you're only at about 30% of the total pressure at that level. Most of the air is below you at that point. Okay, so how could we actually calculate what the partial pressure of oxygen is up at the top of Everest? Okay, well, we just take the total pressure, just up there is like 230 millimeters of mercury instead of 760. And we do the same calculation, multiply by 21%. The proportion of oxygen and nitrogen is the same, both up at Everest and down. Okay, still an even mix. Those two molecules are fairly close in weight, so they tend to stay together, and we've got weather to mix stuff up. Okay, so 21% times 230 millimeters of mercury. Now we're down to 48 millimeters of mercury of partial pressure of oxygen. This is pretty low. Okay, this is basically the level of partial pressure of oxygen that you'll get in metabolically active tissues. This is basically the level where you are at that point. You're trying to your hemoglobin, and we'll see the binding and unbinding curve for hemoglobin. It's not really designed to be able to effectively move from a 48 millimeter of mercury environment to like the 40 millimeter of mercury partial pressure environment of exercising tissues. So it's quite dangerous to stay up there for an extended period of time, and very difficult. It's really at the limit of trained human abilities. Very many people can summit Everest these days, but almost all of them use supplemental oxygen that they're breathing on the way up. Okay. There's a few greats that have trained their bodies to be able to get there without oxygen, but it's very rare. Okay, so let's sort of think about respiration again. And there's really two aspects to the term respiration. Okay, we have external respiration, and we've got internal respiration. So external respiration is referring really to the exchange of gases with the environment into the body itself. Okay, so how are we going to do that? So we're going from the atmosphere, and we're going to exchange between the atmosphere and the air sacs in the lungs, which are called alveoli. Okay, and then those air sacs are then going to exchange with the pulmonary circulation within the blood itself. Okay, within the blood, there's going to also be transport of oxygen and carbon dioxide around the body, between the lungs to the heart to the other tissues, right? And then there's going to be furthermore a delivery and exchange of carbon dioxide and oxygen into the tissue cells itself. So this is where internal respiration is taking place. Okay, so internal respiration is the exchange or the transformation of oxygen plus a food source into carbon dioxide, water, and ATP. So this is how we're generating our energy within our tissues, right? We're burning glucose by combining it with oxygen that's producing carbon dioxide, water, and ATP. Okay. So we have to have an excess amount of O2 that's getting delivered to the tissue cells, and we've got to get rid of that carbon dioxide that's produced by exchanging it into the blood and then transporting it out to the lungs to exchange back to the atmosphere. So what are the lungs? They're really just an infolding of the body surface. Okay, so the circulatory system is going to transport gases between the lungs and the rest of the body, but the lungs themselves and the size and the complexity of lungs is really going to correlate with an animal's metabolic rate. Okay, so a simple lungfish that can flop out onto the cold-blooded creature that sort of flops out onto the turf and onto the ground from the water might have very sort of simple lungs, but we have fairly complex lungs because we have very high metabolic rates. Okay, and let's see. So let's see how those look, what that looks like. So we've got two major divisions of the respiratory system. We have this upper division here and a lower division. In the upper division, this upper respiratory tract, we have the nose, okay, where air is going to come into, pass through a series of filters of hairs into the pharynx, which is this sort of upper chamber here, and then past the larynx right here. Okay, now we also have a glottis and epiglottis, which can direct basically air into the trachea and then direct food down into the esophagus, right? If that, the motion of that particular muscle is disrupted because maybe you are getting conflicting signals when you're chewing and swallowing and trying to breathe at the same time, you misdirect the food down into the trachea, you're going to have choking, right? So we've got to somehow expel that. So that's sort of the role of the pharynx, is to direct it down into the larynx and trachea. Now, the lower respiratory tract consists of basically a series of tubes that continue to branch and branch and branch. At the beginning, they're rigid, and then they become flexible at the very sort of terminal ends of them. Okay, so we have the trachea right here, which is this also known as the windpipe, which is a very sort of stiff structure that then extends down into bronchi and bronchioles, and then finally into the lungs themselves. Okay? So this is where the very terminal ends in the alveoli of the lungs is where the oxygen is going to exchange with the blood, and carbon dioxide is going to come out of the blood into the air. So let's look at a little bit closer at the trachea itself. Okay. So the trachea is basically it's like a little 5-inch tube that's going to connect the larynx to the bronchial tree. So we've got these rings of cartilage, and you can feel them in your throat themselves right here, right? So you have these stiff rings of cartilage that are connected and embedded with connective tissue between them. Oh, this just went out. I got a little paper clip, I guess. Let's see if that works. That works. Uh-oh. Okay. Need to turn it like that, I think. Well, I don't know. I might just turn it off. Yeah. Yes. So I updated the slides. If you downloaded them before, re-download them. They're there. Sorry, I meant to mention that at the beginning. Okay. So this is the trachea. Okay. So why do we want it stiff? Okay. Yeah. Well, is it not? Okay. Well, let me see. Maybe the page is cached. Let's check. Double check. Oop. Okay, that's too bad. Okay, let me let's delete that. Okay, maybe I needed to rename the file or something. Okay, let me upload that, re-upload that, upload files. I think this should be it. Okay, well, hopefully that will update appropriately. Let's see. Okay. That happened. Now that it's totally gone. Okay. Okay, so we've got, well, that's updating. So we want to keep that stiffness present because we want to be able to maintain a pressure gradient in between the body's pressure and within the air pipe itself. Because in order for oxygen or in order for gas to move, it's going to have to be moving down a pressure gradient. Okay, so let's hit upload here or add. And okay, I'm going to just double check that it's the full thing. And Well, it's still doing a preview. Let me know. I uploaded it. I've just re-uploaded it, and let me know if it's still wrong. If it's still wrong, I'll fix it in the class. Okay. So okay. So where were we? Let's get back to here. Okay. Now, so these are going to support and provide a connecting structure between the pharynx and the bronchi, and basically this material can also be sort of bypassed, or the pharynx itself can be bypassed if you have some oral cancer and often common like in cigarette smoking, where you can drill a hole in the trachea and get access to the lungs. This also can happen in an emergency tracheotomy if there's a choking that's occurring that cannot be expelled. Then you might see this maybe in a hospital setting. So let's look now as we pass down the trachea, we get down into this sort of bifurcated structure here where we have a chain of branches that continue to branch and branch and branch. So the trachea is going to split into what are known as bronchi, okay, which each lead to a lung. And then those are primary bronchi are going to divide into secondary bronchi, into tertiary bronchi, and then bronchioles and terminal bronchioles, okay? So it just gets finer and finer and finer until at the end of the sort of bed here, or at the end of these sort of terminal bronchioles, we have the alveoli that look like little grape sacks essentially on sticks, okay? And this is where there's actually going to be quite a bit of elasticity and volume changes within the structure. On the way down here, these are all pretty stiff, okay? So you're not going to get large changes in structure, but once you get down to the alveoli, this is where we're going to see an expansion and contraction, where the major changes in lung volume take place. So how does the lungs actually inflate, deflate? Well, they are going to basically fill up this area called the rib cage, right? That extends from our diaphragm down at the bottom of the rib cage all the way up to our collarbones up here. Okay. Now the lungs are not exactly identical. Okay, they're similar to one another. Their contents are similar, but the overall gross anatomy is a little bit different. We've got sort of three lobes, and it's the right lung has got 3 lobes and it's shorter and wider. And then the left lung, it's got 2 lobes and it's got a little cutout here, which is the cardiac notch that sort of gives room for the heart to exist. Okay. Now, surrounding these lungs, okay, is a pleural sac, which is this fluid filled sac that allows the lungs to expand and contract without tearing the tissues. Okay, so the alveoli are very delicate. Okay, and they need to be protected. Okay, and so just sort of yanking on them with connective tissue could potentially cause them to tear. And so instead, we use a combination of surface tension from water and a slight change in pressure between the inside of this fluid-filled sac and the alveoli themselves in order to expand and contract it. We also need, because there's such a large change in volume of the alveoli surface needs to be able to freely smooth in a basically frictionless way across any membrane that is touchy, but still be able to have the ability to be pulled on to expand or relax back down to collapse. So you can sort of imagine the alveoli looking like this little lollipop here that's surrounded by a water-filled balloon. And so this lung, okay, is surrounded by this entire very thin sac. So this is the outer edge of the sac is called the thoracic wall, okay? And then the inner edge is the visceral pleural. Okay. Now, we have the diaphragm down here. This is a muscle that's going to flex to expand or contract the overall cavity size. And one thing to note here that it's a lot thinner than we're actually illustrating here. You know, this is almost like microscopically thin. We have 2 sides to the pleura. We've got the visceral pleura stuck to the lungs and the parietal pleura, which is stuck to the thoracic cavity. And this fluid inside is going to be secreted by surfaces of the pleura, and it's going to lubricate these surfaces that are going to allow it to freely slide against one another while still sticking to the lungs. Okay. Now, you can have an inflammation of the pleura, and this is called pleurisy, and that can lead to difficulty breathing and pain when breathing. Now, the overall location of these lungs are in this rigid thoracic cavity. Okay, so this is the thoracic cavity is essentially the rib cage and the surrounding structures around the rib cage. And the lungs are, of course, smaller than that thoracic cavity, but they're very elastic. They can stretch out. And normally they're stretched under normal conditions. Okay, so this is our lung. Here's our pleural sac again. And you can see that there's actually a pressure differential between the thoracic cavity, between the pleura, and between the lungs. Okay. Now, in order to have the lungs inflate, you have to have a negative pressure gradient relative to the outer airway. Okay. And how are we going to achieve that negative pressure gradient relative to the atmosphere? Well, we've got to sort of expand the lungs themselves. We've got to pull on the lungs somehow. And we do that through a negative pressure gradient between the pleural sac and the lungs themselves. So this negative pressure gradient is sort of established by the elasticity of the pleural sac, which is sort of pulling we can sort of pull the pleural sac a little bit apart, and that's going to drop the pressure inside the sac. Okay, and the overall differences in pressure here are actually quite small, though. We're only talking about like 4 millimeters of mercury difference between inside of the pleura and the thoracic cavity, or the inside of the lungs. Okay, so this differential here is called the transmural pressure gradient. Okay, so you can measure that across the lung wall here. So which would be like the interalveolar pressure minus the interpleural pressure, or we can measure it across the thoracic wall, and that has a very similar gradient. So the pleura is always here at a lower pressure than both the alveoli and the thoracic pressure. Now, what will happen is that the thoracic cavity here is stuck to this parietal pleura because of that transmural pressure gradient. And if we expand the chest cavity, that is going to cause a pressure drop within the pleura and cause an expansion of the lungs. Okay, so so what will happen if something goes wrong? If we get a stabbing or gunshot wound, or maybe you fell off a rock and you're climbing and you damage a lung and you collapse the lung. Okay, how does that happen? The collapsed lung is called a pneumothorax, and there's really two ways that you can do it. First, it could be from a trauma where you puncture you have some wound in your chest where you puncture the outer pleura here, the outer pleural sac, and allow the pressure from the thoracic cavity to normalize, to equalize with the pleura. In the absence of a pressure gradient now across the pleura and the atmospheric pressure within the lungs itself, the lung is going to collapse. Okay? So the lung needs to be surrounded by a slightly lower pressure in the pleural sac in order for it to maintain its size and expansion. Okay? So this is going to cause a collapsed lung. The same thing can occur if you create a hole in the other side. Okay? So this could be spontaneous pneumothorax. This could be potentially due to some pathogen like that causes pneumonia, for example, where you get some opening of some pores that open within the lungs into the pleural sac, allow mixing of fluid and pressure between that, and that will also result in a collapsed lung, even though the thoracic pressure is so it'll allow a normalization of the lung and the pleural sac pressure and cause a collapse as well. Okay, so where does the gas exchange actually occur? It's going to occur between the atmosphere and the alveoli, and so the air is going to come down through the bronchi to the bronchioles, and then eventually into these alveoli. And you can see here that sort of each terminal bronchiole is going to branch off into these little clusters of alveoli. Okay, and the clusters each cluster is going to have its own pulmonary artery and its own pulmonary vein that's going to feed the blood flow into and out of it. Now, the blood doesn't go directly into the alveoli. it goes in a meshwork of capillaries that closely surround these sacs. Okay? And if we zoom in here, you can sort of see where the gas exchange occurs. So we've got the alveolus here. That's the sort of term for a singular alveoli. Okay? And this is where the air is going to come in. Okay? We have a respiratory membrane that is extremely thin, and that is where the gas exchange is going to pass through there to enter the bloodstream. Okay, so oxygen is going to come into the bloodstream here. Carbon dioxide is going to exit the bloodstream. And these processes are going to be driven by diffusion because they're over very small distances. We're talking micron scale distances here. The alveoli have very thin membranes that are one cell layer thick. So do the capillaries, right? So that's going to provide sufficiently small distance for diffusion to work. Now, the cell membrane is moist. Okay? The alveoli here is moist, which is important for having that gas exchange, but it also has a problem that needs to be overcome in order for the lungs to maintain inflation. Water is actually, it's a good lubricant, but it also has some stickiness to it in itself. Water tends to want to stick together. That you can see this with surface tension, right? Surface tension in a glass, you keep filling water up in a glass, and you can maybe get the surface of the water a few millimeters above the edge of the glass if you're very careful because of that surface tension. So it's, it doesn't have a huge surface tension, doesn't have a huge effect at like macroscopic scales, but at microscopic scales like this, it's actually quite powerful and quite a powerful force. Okay? And that surface tension is going to tend to try to pull the edges of the alveolus together. Okay, it's going to try to collapse the alveoli or the alveoli, and so there has to be some way to prevent that from occurring. Okay? In order to prevent that from occurring, we have a surfactant that is produced. So a surfactant is basically just a soap. And this is it's an antiempathic molecule. That means it has a polar side and a nonpolar side. It's going to be able to break up the surface tension that's present along this surface here. Okay, so you can imagine all the water along the surface, this moist surface. You can also imagine the water molecules along an alveolus sort of pulling on each other and trying to collapse that. But by inserting a few molecules of this surfactant, that's going to break up those pulling forces, and it's going to really reduce the surface tension. So this discovery that there are these cells here, these are called septal cells, that are going to release that surfactant. That's their sort of primary job, is to keep the lungs a little bit soapy. That discovery was instrumental and critical for extending the period of time at which premature babies can be saved. Okay, so basically, once a surfactant was discovered, which I believe the surfactant sort of production comes online around 28 weeks. Okay, you have to apply a sort of postnatal 28 or post-conception, about 28 weeks post-conception. So a premature baby born around that point needs to have artificial surfactant delivered into the lungs and to allow those lungs to inflate. Otherwise, they'll never be able to take a breath because the surface tension is too high. So that is a medical intervention that has saved many, many preterm infants. So there's another factor that allows the alveoli to prevent collapse from this surface tension, and that's alveolar interdependence. So a single alveolus is generally not alone. It's in a cluster, okay? And it's surrounded by other alveoli. Okay? So, and you can imagine that if each one of these, let's just think about one alveoli here, okay? And the surface tension is pulling it in, okay? Starting to collapse. What will happen? If it's stuck to these other ones, that's going to tend to pull the other ones outward towards it, right? So the more that this one collapses, the greater the force that's pulling and distending these other alveoli. Okay, so in practice, okay, all of these alveoli together are trying to collapse on themselves, but since they're stuck together, they sort of have a counteracting force on each other that sort of keeps them in balance and acts as a tug of war that's going to maintain their overall size. So to put this all together, we've got some forces that are keeping the alveoli open. That's going to be the pressure gradient, the transmural pressure gradient between the pleural sac and the lungs themselves. We've got this surfactant, the pulmonary surfactant, which breaks down the surface tension. And we have this interdependence of the alveoli. If one collapses, it opens up and pulls on the other ones to keep them to increase their size. Then on the other hand, we have some forces that are actually trying to create that collapse. And that is going to be stretched tissues that surround it. So the pulmonary connective tissue fibers, like the stuff that is surrounding the capillaries, those tend to want to try to stick together and collapse the alveoli, as well as the surface tension. The surface tension is reduced by the surfactant, but it's not zero. Okay. So these two forces are sort of in tension with one another, or these forces are in tension with one another. And by essentially controlling the transmural pressure gradient, we can effectively inflate or deflate our lungs. So how does inflation and exhalation work? This is all going to be controlled by muscles within the thoracic cavity system. So the inhalation itself is going to be an active process. Okay? We're going to expend energy to contract particular muscles that will expand the thoracic cavity, increase the pressure differential between the thoracic cavity and the pleural sac, and thus also the pressure differential between the pleural sac and the lungs. So when we inhale, okay, we have a few different things that are going to contract. We have the diaphragm down here. This is like a little dome-shaped muscle that sits below our lungs. And when it contracts, it straightens out, okay? So it's sort of pointed up, and when it contracts, it pulls and straightens out and opens up the cavity from below, okay? That's going to cause a drop in pressure and will cause the lungs to expand to fill the space. Okay? On the other hand, to exhale, generally, exhalation is going to be passive. All we've got to do is just relax, and the air is going to naturally flow out as this sort of as the thoracic pressure sort of pushes down against the lungs. Lungs will then expel the air out. So the diaphragm is not the only muscle that's in play here. We also have these external intercostal muscles. So in between each rib, okay, we have these both external and internal intercostal muscles, and they can contract in order to raise the rib cage or drop the rib cage. Okay, so when we inhale, our diaphragm is going to contract the diaphragm, increasing the bottom size, as well as we get a contraction of these external intercostal muscles that are going to bring these ribs together and pull them upwards towards the collarbone. That's going to now increase the space on the top and sides, horizontal sides, that are going to allow a greater sort of volume for the lungs to expand into. Okay. So when that lifts up, it's sort of like, I guess you could think of it like the lid of a bucket lifting up, where the rib cage and the sternum, which is this hard piece that protects the heart, are all going to lift up together, expanding that thoracic cavity. So what about the expiration? All we have to do to breathe out is to relax that diaphragm. It's going to snap back, relax those external intercostal muscles, and that's going to lower the rib cage down. It's going to reduce the amount of volume there, and it's going to expel the air. That is in our normal sort of involuntary breathing. But sometimes we want to really breathe harder. We want to expel the air faster, say if we're exercising, and we're actually going to then do contractions of distinct muscles in order to accelerate that pace of and change the overall amount of volume that we can exchange. So in order to do that, we're going to contract these internal intercostal muscles, which are sort of on the inside of the external muscles. And that contraction of those muscles will actually bring the rib cage down, decreasing the volume. And we can also contract our abdominal muscles, which will sort of also push down and pull those rib cage down in order to further expel the lungs. Okay, so this is an active. So exhalation can be passive, which is in the normal case, or active to really cause the biggest change in volume. Okay. So okay, and so now let's look and see how that breathing is actually regulated by components of our bloodstream. How do we know when to breathe? How does our body sort of figure it out, or what pace we should be breathing at? It's largely an involuntary process. And it's really driven by the presence of carbon dioxide, okay, in our blood. Okay, so last time we talked about what are the components of blood. Two of them, we've got water as a major component, is the primary component of blood. But some of the other things in blood that are quite important is bicarbonate and protons. So bicarbonate and a proton can basically is a dissociated form of carbonic acid. So these are sort of in balance with one another. Okay, and then this carbonic acid can transform into CO2 and water. Okay. Now, CO2 and water, like carbon dioxide, is a pretty stable molecule. Usually it's not going to break down by itself. So there's actually an incredibly important enzyme within our body and within all animals called carbonic anhydrase. This is going to lower the transition energy for carbon dioxide to combine with water to produce carbonic acid dramatically. So this reaction is now incredibly fast, and the more carbon dioxide that you produce within a body tissue, that will immediately get transformed into carbonic acid, okay, which can then break up into this proton and the bicarbonate. Bicarbonate sort of acts as a buffer for the protons to maintain an even pH. But the more carbon dioxide you produce, that's going to tend to push the reaction in this direction, going to produce more protons, more hydrogen ions, which is then going to decrease your pH. So carbonic acid is going to be a way to transport carbon dioxide around, and it is a form of. That's a good question. Why do we have carbonic acid? I think it's really for transportation around and the ability to move carbon dioxide to different places within your body. Okay, so here so how does this loop sort of work? If we have a high level of metabolism in some tissue, what will happen is we're going to deplete the amount of oxygen in an area, right? So we're going to get a drop in the partial pressure of oxygen, and we're going to increase the partial pressure of CO2. That is going to then cause a decrease in pH because of an increase in protons. And that is going to be sensed by chemosensitive cells within the brainstem, which will then drive an increase in respiratory rate. That will tend to counteract this drop in O2 and the increase in CO2 by increasing the rate of gas exchange with the environment. Okay. So this sort of, we also can have some feedback here where if the oxygen levels get really, really low, they also can sort of directly affect the brainstem respiratory center. But mostly, the control of the respiratory rate is going to be dependent on the overall concentration of carbon dioxide. So where in the brainstem is that? It's in the medulla oblongata and pons in the brainstem. Those are the control centers. And these are involuntary control centers. This is not something that, this is not the muscles that we are driving when we're consciously trying to expel, or it's not the centers that we're driving when we're consciously trying to decide to breathe really hard or hold our breath or things like that. Instead, it's just a rhythm that's set up by electrical activity in the pons and the medulla oblongata, and it sends out, and it's going to tend to cause the diaphragm to contract for about 2 seconds, relax for 3 seconds. That's going to set our standard respiratory rate. Okay. You can override it using higher brain functions to change that voluntarily, but you can't completely override it, right? If you try to hold your breath for a really long time, eventually you're going to start to panic. You're going to feel extreme distress, and you are going to involuntarily breathe. Okay. So this is because of the buildup of carbon dioxide in your blood and the dropping of oxygen. So there are chemoreceptors in your carotid and aortic arteries, and they are going to basically feed back and send messages to the brainstem that will cause you to trigger that increase in respiratory rate. Now you can train yourself to suppress that over long periods of time. Lots of training. You can train yourself to improve your oxygen handling and oxygen sort of saturation of your muscle tissues over repeated training. This is what, say, deep sea divers do, pearl hunters, or these people that are trying to set records. You can train that, but in general, this is how it's going to work. Okay, so this is sort of another way that you can visualize that. Okay, so what about the lungs themselves? How much volume are we exchanging? And what are the sort of technical terms here that you should know if you want to understand how lung function works? So the recording of lung volume is called spirogram, okay? And we have a few different terms here. First, we have the tidal volume, okay? So the tidal volume is the lungs function basically as a tidal system where something comes in and comes out through the same tube, okay? Much like the waves, the tides come up and down on the same sort of beach, okay? So during normal respiration, our tidal volume is maybe half a liter, okay? Just going back and forth, back and forth, back and forth here between a involuntary exhalation and inhalation peaks of those things. Now, the inspiratory reserve volume is this sort of peak here, which is how much air can you breathe if you do maximum voluntary inhalation. And it's considerably more. If you try to hold your breath, right? If it's considerably more than the normal amount that you're going to breathe during rest. Now, as you're exercising, this tidal volume is going to increase up to that inspiratory reserve volume. If your respiratory rate is going up and the amount of contraction that you're doing is going up. And we also have the term the expiratory reserve volume, which is how far down you can go with a maximum effort here. And even if you fully try to expel all of your air, there's still going to be some leftover volume. You can't completely collapse the lungs. So that residual volume down here is the remaining amount of sort of volume that is still present. So that residual volume limits actually the overall efficiency of gas exchange, right? Because you're always going to, any air that's coming in, fresh oxygenated air that's coming in is always going to get mixed with that remaining residual volume of air that is sort of saturated with carbon dioxide and is depleted with from oxygen. Okay, then so our total lung capacity is the maximum amount of volume that the lungs can hold. Our inspiratory capacity is how much we can inspire after a normal expiration. So from here, or wait, inspiratory capacity is from here to here. And then our functional residual capacity is the volume of lungs at the end of a normal expiration. So that's from here to here. And then the vital capacity is the amount that you can basically breathe in from maximum exhalation to maximum inhalation. Okay, so that's basically how these volumes are going to change. What happens to the partial pressures of carbon dioxide and CO2 as the air is inhaled and then passed to all the different tissues through the circulatory system? Okay, so let's take a look here. Our inhaled air, okay, is going to have, at atmospheric pressure, is going to have 160 millimeters of mercury of PO2 and about 0.2 millimeters of mercury partial pressure of CO2 up here at the top. Okay. Now, gases are going to want to move down their pressure gradients, again, from higher to lower pressure. As the air comes into the alveolar spaces, okay, it is going to mix with that residual air, that residual volume that was in there. Okay. So that's going to tend to decrease the overall level of oxygen and increase the overall level of PCO2. Okay. From there, it's going to exchange and equilibrate with the alveolar capillaries. Okay. So as soon as you inhale the air, within a few seconds, you get a complete exchange, an equilibration of what's in this inhaled air mixed with the residual air into the alveolar capillaries. So so now this is going to get returned by the pulmonary veins back to the heart, okay, and then pumped out the systemic arteries, and nothing changes in that partial pressure of both oxygen and carbon dioxide at that point. Okay? It's fixed because there is no gas exchange there, right? They're thick tissues, diffusion's not going to work, the blood is moving quickly. The only exchange that's going to take place is once you get into capillary beds. Okay, so sure, there's some capillary beds on the heart surface, but the primary areas of the gas exchange is going to be in the systemic capillaries of the circulation near to any tissue. When we get there, we're going to unload our oxygen and we're going to load up carbon dioxide, because the body tissues themselves will tend to have an oxygen level that is less than the oxygen level in the systemic capillaries, and will have a higher concentration of CO2 than in the systemic capillaries. That's then going to return to the heart, to the right side of the heart, okay, where it will stay steady until we get up into the alveolar capillaries of the pulmonary circulation, in which case it's then going to exchange with the inhaled air, and the exhaled air will be have a much greater level of carbon dioxide in it, okay, and a somewhat less amount of oxygen relative to the inhaled air. Yeah.
Does the heart need its own like blood supply, because it like can it not do like can like the heart cells not get oxygen through?
Yes.
Like
yes. So the heart muscle wall is really quite thick. The fusion's not going to work. So you have your coronary arteries. You've got 4 of those that are going around to each one of the chambers of the heart, and you need to have you have a very dense capillary network there. Where's the slide on this? I think maybe 15. Here, I'll show you. Right there. It's kind of hard to see, but you can see here. So here is, here's our aorta here. Here's a coronary artery, okay? And then the heart muscle itself has intensely vascularized capillary beds, because the heart is a highly metabolically active tissue. So you do have to have a lot of blood being delivered to that. But that's not driven, that doesn't just happen through the wall. It has to happen through a very dense network of capillaries into the it's basically a component of the systemic circulation.
Can the heart start doing fermentation if it doesn't have oxygen?
Can it start doing what? Fermentation?
Yeah. Is that
I think that is not going to provide nearly enough energy for it to continue to beat. Okay, so if you have a blockage in a coronary artery, right, then you cut off oxygen supply to the heart. That's going to cause that is a heart attack. Okay. So let's go back to the slide. Okay, so here's so we're basically extracting some amount of oxygen from the atmosphere, and we're putting out a bunch of carbon dioxide back into the atmosphere, and we are transporting it around through the blood here. Now, here's the thing about blood and about water. It is a terrible medium of oxygen transportation. Okay, it is not good enough for basically for any warm-blooded creature that has a high metabolically active rate. You are never going to just use the solubility of oxygen in water or in a fluid itself to carry the oxygen around. You just cannot get a high enough density of oxygen into the water. Let's look at this. So at at sea level, a partial pressure of oxygen in the air is about 160 millimeters of mercury. That's the same if it's in water. Same partial pressure. Okay. Okay. The amount of oxygen that you can fit into 1 liter of air, okay, at that partial pressure is 210 milliliters per liter. By definition, it's 20% of the liter of air. Okay. In water, water's pretty dense, much denser than air, right? Like literally like almost 770 times denser than air. Okay. And so there's much less space for the oxygen to be able to dissolve into the spaces between the water molecules. So the concentration of oxygen that can be dissolved into water at the same partial pressure is about 30 times less. Okay. So that makes it really crappy, right, for being able to deliver oxygen by itself. Also, water has a much higher viscosity, so it's harder to move than oxygen, or harder to move than air. right? So that like leads to this design challenge that the body has to solve, and most animals have to solve, which is how are we going to efficiently transport oxygen within our tissues? Okay. And that is where respiratory pigments come in. Okay, so respiratory pigments are like proteins that are going to transport oxygen around. They dramatically increase the carrying capacity of oxygen in the blood. And different creatures have different respiratory pigments. You've got arthropods, mollusks. They use hemocyanin, okay, which is basically uses a copper ion that binds the oxygen. Most of our vertebrates and some invertebrates use hemoglobin, okay? And the hemoglobin has an iron core to it that iron can coordinate the binding of oxygen. And it has a quaternary structure here. It's a sort of a tetramer here. And each one of the four components can bind a single oxygen. Okay. When you bind the oxygen, it's going to change the conformational state of the oxygen, or change the conformational state of the hemoglobin to enhance the ability of other binding sites to bind to oxygen. So there's a cooperativity in hemoglobin to oxygen binding. Now, these respiratory pigments in humans are carried in red blood cells, right? So we talked a little bit about red blood cells last time, okay? So here's our little capillary, here's our basement membrane, and here are some tissue cells, the endothelial cells that line the capillary, and then our red blood cells are getting passed tightly through this capillary bed, okay? So the hemoglobin, the red blood cells are these enucleated cells, no nucleus in there. They've got about 200 million hemoglobin molecules per cell. Okay, and we've got 4 atoms of iron bound to each one. So, you know, each one of them is containing, each red blood cell contains almost a billion, can hold up to maybe a billion or 800 million oxygen molecules. Okay. And the hemoglobin has particular binding characteristics where oxygen is high, it tends to hang on to it better, and then it's going to release oxygen in areas where the partial pressure of oxygen is less. Okay. How do we make red blood cells? They are formed from stem cells that then turn into myeloid stem cells. These are our pluripotent stem cells, which turn into myeloid stem cells in the bone marrow, then turn into these reticulocytes, which then get rid of the nucleus, which then finally matured into a erythrocyte or a red blood cell. Now, since they don't have a nucleus, they don't have any protein production machinery, they cannot repair themselves when damaged. So there's just these sacs, and they're going to circulate around your body for about 120 days before they finally are broken down sufficiently to no longer be able to carry the hemoglobin. And that's going to be, that breakdown process is going to happen primarily in the spleen, somewhat in the liver, and they're going to recycle the hemoglobin that is within those and repack it into new red blood cells. Okay. We break down an insane number of these per second, like 2 million red blood cells get broken down every second in our body. We've got to remake those 2 million red blood cells all the time, right? How do we make red blood cells? We're going to control that rate based on hormones. If we have low blood oxygen level, that's going to stimulate the kidneys to create a protein called erythropoietin, which is a hormone that is then going to travel to the bone marrow and tell the bone marrow to upregulate the production of red blood cells. Okay. So we can so there's different ways that you can get that. You can, if you're trying to train for a race and to have greater aerobic endurance, you want to have more red blood cells, you can go try to train, maybe not on Mount Everest, but maybe in the mountains of Colombia or somewhere, some really high place where you're going to have a lower oxygen level, and that'll tend to upregulate your red blood cell production. Or you could use blood infusions or EPO, erythropoietin. That'll increase your percentage of red blood cells in your bloodstream. Okay, so how are these hemoglobin molecules actually transporting oxygen around? Okay, so we need them to concentrate the oxygen because the blood itself cannot dissolve sufficient oxygen into it to meet our metabolic needs, right? And so we have a reversible binding of oxygen to hemoglobin. When we have very low partial pressure of oxygen, we're going to release oxygen from hemoglobin. At high partial pressures of oxygen, it's going to bind to hemoglobin. Okay, we only have 4 slots per hemoglobin, and it doesn't matter if you go to 200 or 300 or 400 millimeters of mercury of partial pressure of oxygen, you are not going to add any more oxygen to the hemoglobin because there's only 4 spots. So at 100 millimeters of mercury, which is about the level that is, in that inhaled air that's been mixed around, you're going to get full saturation of your hemoglobin. Okay. As you circulate through the bloodstream and go to areas that have lower partial pressure of oxygen, you're going to release that. So the goal here is to saturate oxygen levels when we're at the partial pressure that's in the lungs, okay, and then release the oxygen at the partial pressure of the tissues. We're basically just moving the oxygen from high PO2 areas to low PO2 areas. So we have to have binding characteristics of hemoglobin that will satisfy that particular functional goal. All right, so let's take a look and see how that works. Here's another plot. First thing that we need to look at our plot is, what are we even looking at here? Okay. On the x-axis, we've got the partial pressure of oxygen in millimeters of mercury. Okay, and we're going from 0 to 100 here. Okay, so in the lungs, we're at 100, maybe 104 millimeters of mercury at atmospheric pressure. Okay. And here is the saturation of hemoglobin. That is, what percentage of the binding sites on the hemoglobin are bound to oxygen. Okay, so at 100 here, that would mean all 4 sites are bound. Right? At 0, that would mean 0 sites are bound. At 50% here, 2 of the 4 sites would be bound. And when we're up at 100 millimeters of mercury PO2, we're at about 97.5% of these sites are bound. And it really, through a fairly large range here, there's not a big change, right? So, okay, if we put one up to 200 millimeters of mercury, maybe we could get up to 99% bound. But that's not really increasing the amount of oxygen that's being bound to the hemoglobin very substantially. I want to compare this curve now, to this curve down here, which is basically a flat line, a linear line here, of how much oxygen is dissolved in the blood itself in the absence of the red blood cells. Okay, just in the fluid itself, in the plasma. There is a little bit, but it's not very much, and it's not going to be sufficient to meet the metabolic needs. Right. So as we sort of circulate through from our lungs, the alveolar sort of capillaries, into our heart and to our systemic circulation, we're always up here at 100 millimeters of mercury of PO2. It's only when we get down into the tissues where the partial pressure of oxygen begins to drop. Okay. So, okay. So let's take a look here. So we're in the pulmonary capillaries. Oxygen is loaded up here. We get close to 100% saturation. Okay. And then if the oxygen level in these capillaries falls a little bit, okay, let's say maybe we're at a higher altitude, so we have less partial pressure of oxygen, we still can get, through a fairly large range of altitude or of partial pressures, we still can get almost complete saturation of the hemoglobin. Now, as we move down to lower levels of PO2, we're going to start to unbind oxygen. Okay. So this steep portion of the curve here is going to occur in the systemic capillaries where oxygen is being delivered to the tissues. Okay, so in the tissues where you don't have a whole lot of metabolic activity, you're going to be maybe up here at 40 millimeters of mercury of PO2, and you'll unbind maybe 25% of the oxygen there. You'll be at 75% saturation. That means 3 of your sites are still bound, 1 of your oxygens fell off. Okay. If you go into highly metabolically active tissues, like your legs that are, if you're running, okay, you might get down to 20 millimeters of mercury of PO2. And what's going to happen there? Now you're going to only have about 1 molecule bound. So there's going to be a selective release of oxygen two tissues that have the greatest metabolic loads because of the difference in partial pressure of oxygen within those different areas. Okay. So when we move from highly metabolic or low metabolically active tissues to high metabolically active tissues, we get a huge drop in that saturation level. So a selective unbinding. And then let's see here. Now, there are a few things that can also further affect this curve and which can continue to direct oxygen to be released into areas that are most important or most needed. Okay. So one thing that can affect the binding ability of oxygen is temperature. Okay. So at lower temperatures, the affinity of hemoglobin for oxygen is increased. Okay. Now, where in the body might have a lower temperature than a metabolically active like a muscle? Now, when you have metabolically active muscle, your mitochondria are making ATP. You're going to have a much higher temperature there. That is where, basically the heat in your body is coming from. So those tissues are going to be hotter. Okay. The lungs, though, you just exchange air with the environment, right? So the temperature of air within your lungs is considerably lower than the rest of your body, your core temperature, and even lower and that in turn is lower than the most metabolically active areas. Okay, so that effect here will cause a greater saturation of oxygen in low temperature areas like the lungs, and it will cause a release in a lower affinity and a release of oxygen in areas of higher temperature. So I mean, you can look at it right here. So imagine you're right here on this curve. Okay, if we're at 37, body temperature here at the same partial pressure of oxygen, okay. If we're at 37 C, then we're at 75% bound. If we're at 33 C, say in our lungs, okay, then we're like 82% bound. If we're in a hot tissue, 41 degrees C, we're at 62%. So there is a pretty big range here where oxygen will tend to be released in areas that are hotter, and it will tend to be bound to the hemoglobin in areas that are cooler. Yes.
And like sitting the air that comes in, like you said, that's a purpose of the upper respiratory tract. It's better.
I mean, I think it's simply a consequence of, the moistening is very important for getting any gas exchange to take place. You can't do it through a dry surface. Oxygen will have a hard time passing through that. And then heating up, I think probably just to reduce the overall temperature differential. I don't even know if heating up is like it's more of a effect that's going to happen, but it's still never going to reach the total. It's still going to be at a lower temperature than what's going on inside your body. Okay. So temperature can affect hemoglobin affinity, but there's other factors too. CO2 and pH actually has a pretty important impact on affinity of oxygen, of hemoglobin for oxygen as well. So in a low partial pressure of CO2 or high pH environment, we're going to shift the curve to the left. In a high PCO2 or low pH environment, we're going to shift the curve to the right. Okay, so basically, the more acidic you are, okay, then the higher carbon dioxide area you're going to have, the more that this curve is going to travel in this direction. When you're shifting the curve to the right, that means you're lowering the affinity for oxygen at a particular partial pressure of oxygen. So even if you have the same partial pressure of oxygen in here, if you're at a pH of 7.2, you have way less bound than at a pH of 7.6. So pH itself has a profound impact on the delivery of oxygen. And where is the pH lowest? It's going to be in areas where we're producing the most carbon dioxide, because the carbon dioxide that gets turned to carbonic acid, which then gets turned into protons and bicarbonate, right? So that's another factor now that is going to selectively release oxygen in the highly metabolically active tissues. And this is called the Bohr effect, this sort of pH effect, which was discovered by the father of Niels Bohr, the famous atomic theoretician. Okay. And yeah, that's just a reminder of how that flow takes place. Yeah, so increased CO2 levels, you're going to get a rightward shift in the curve because you're going to increase protons and decrease pH. Now, okay, so we just looked at that, right? Okay. Okay, so, and this is sort of like how, so there's another effect of this, okay? Now, this is actually caused by, I guess, how the reason that this takes place, okay, even if you have a fixed level of carbon, okay, I guess what I should say is there's two factors here, okay, that cause this to take place. One is a direct effect of the protons on the hemoglobin, and two is an effect of the carbon dioxide on the hemoglobin. So the first effect, the pH, is because the hemoglobin can get protonated. So various amino acids on the surface of the hemoglobin can get protonated, and a proton can bind onto some of the sort of free electrons floating around there. And that changes the affinity of the hemoglobin itself for oxygen. It reduces it. Okay. So if we have the same level of CO2 here, but we control the pH, okay, then we get a rightward shift with decreasing pH. Yeah. But what happens if we hold the pH constant and change the carbon dioxide levels? It turns out that the carbon dioxide can also bind to the hemoglobin and turn into carbaminohemoglobin. Yeah. So that is going to also change the conformation state of hemoglobin to make it less likely to bind onto oxygen and release the oxygen. And in fact, a major way that carbon dioxide is transported in the body is through these carbaminohemoglobin binding. Okay, so carbon dioxide can get transported just sort of dissolved in the blood. It can get transported as carbonic acid, and it also gets transported partially by actually making a covalent bond with hemoglobin and turn it into carbaminohemoglobin. Yeah. Okay. Let's see. There's one other way that we can sort of change the affinity of hemoglobin for oxygen, and that is through a sort of molecule called DPG. So this is sort of bisphosphoglycerate, okay? And this concentration of DPG changes depending on the metabolic load. So if you have a high metabolic rate, then you're going to produce DPG. DPG is going to bind onto hemoglobin and basically displace oxygen from the hemoglobin. The higher the concentration of DPG, the more the rightward shift, and the more you're going to release oxygen into the blood. Okay, so sort of in sum here, the factors that are going to affect oxygen binding, we're going to have a shift to the right, so an unbinding of oxygen at the same PO2 levels in metabolically active tissues because of increased temperature, decreased pH, increased carbon dioxide levels, and increased metabolism. All four of those things together really help direct the oxygen to release in the places it needs it most. So if you have a rightward shift in the curve, you're going to have a decreased oxygen affinity. Leftward shift is an increased oxygen affinity. Overall, for carbon dioxide, about 5 to 7% of it's going to be actually dissolved in the plasma. About 23% of it is going to be bound to these hemoglobins and carbaminohemoglobins, and the rest is transported as bicarbonate or carbonic acid. Okay. For oxygen, only about 2% of it's dissolved in the plasma. 98% is bound to the hemoglobin. Okay. So hemoglobin is important for both oxygen and carbon dioxide transport. Right, and this carbonic anhydrase can convert CO2 to bicarbonate and protons. The CO2 and protons can bind to hemoglobin, displacing oxygen, releasing more oxygen to the areas that need it most. That's the Bohr effect. And we also can decrease the ability of of hemoglobin to bind carbon dioxide and H because of, so if oxygen is bound to hemoglobin, that actually sort of blocks the ability for carbon dioxide to bind to that hemoglobin. And that's called a Haldane effect. Okay. So together, we're going to deliver oxygen to the tissues with the Bohr effect and increase CO2 delivery to the lungs by the Haldane effect. Okay. And I think that's it. Thanks, everybody. Good luck on your exam tomorrow. I hope it goes well. And enjoy spring break. We're going to talk about the immune system next, which is the immune system is the second most interesting system in the body after the brain. So I'm pretty excited about that section. Thank you.